right, my clock says 6:30, which means it is time to start tonight's installment of the Coastal Lecture Series. Good evening, everybody. My name is Dr. Zach Judd. For those of you who haven't attended this series before, I welcome you. We're, as I mentioned, in a little bit of a different venue this evening, and I'll explain that in a second. But first and foremost, I want to thank all of you for attending tonight. I know many of you are probably used to attending at the Blake Library, and I appreciate you pivoting to attend in a new venue tonight because of a water main break. We got a call this afternoon from the Blake Library letting us know that there was a massive water main break on Monterey, and the library was closing down, as well as the county building where all of our Zoom broadcasts originate from. So rather than canceling the entire event tonight, we realized that we already had quite a few people signed up to join us via Zoom. We made a mad dash effort to let the rest of our attendees know through email and social media that we were pivoting to a Zoom only format. And I certainly hope that many of you watching tonight are part of our normal Blake Library audience. And again, I apologize for the inconvenience. We thought this was a better option than going ahead and completely canceling Dr. Paul's presentation for this evening. Now, I do want to thank everybody again for being a part of the series this year. We've had an amazing start to the 2026 Coastal Lecture Series. Last week's lecture had an attendance of 599 participants. Between the Blake Library, we had 245 at the Blake and 355, I think, watching on Zoom. It was just an incredible turnout, and it shows us how important this quest for new knowledge is for all of you. And that's the whole reason that we do the lecture series. So again, we appreciate you being here. Tonight's special guest speaker, Dr. Valerie Paul, is the director and head scientist at the Smithsonian Marine Station at Fort Pierce, a research center that's part of the Smithsonian National Museum of Natural History. Dr. Paul's research focuses on marine chemical ecology, coral reef ecology, interactions between marine plants and grazing animals, marine cyanobacteria, and marine natural products. She holds a PhD from the University of California, San Diego, Scripps Institution of Oceanography, and she's a fellow of the American Association for the Advancement of Science, a Sigma Xi fellow. Her work on coral reefs has been recognized by the Eminence in Research Award from the International Coral Reef Society. And Dr. Paul, incredibly, is the author or co-author of more than 370 scientific research publications. That is an astronomical number. Dr. Paul has been the head scientist at the Smithsonian Marine Station for the last 23 years. And before moving to our side of the globe, she was a professor at the University of Guam Marine Laboratory from 1985 to 2002. Tonight, Dr. Paul will tell us about the complex interactions between plants, animals, and the chemicals that make them tick. It is my greatest pleasure to introduce Dr. Val Paul.
The last time I gave a lecture for FOS was during COVID, and I had to do that by Zoom too. So this time I was actually kind of looking forward to seeing people in person, but so be it. The water main decided to interfere with that. So I'm really glad we could continue with this Zoom format and that we have so many people online. I think it's a great opportunity to still share with you what I wanted to present this evening under the circumstances. Also, the last time I spoke with you, I talked almost entirely about the Smithsonian Marine Station and the research that was going on, a little bit about my work, but more broadly. And this time I thought I would just have fun telling you about some of the research we've been doing for the last, probably 20 years or more, especially the last 20 years since I've been in Florida, or almost 25 now, and back in Florida, and then really share with you some of the ongoing work that we're doing. And I think this, hopefully you'll enjoy it and learn a few things about marine chemical ecology. I want to first start by telling everyone, thanking everyone that has worked with me over the years. I have an amazing team of people at the Marine Station that's up in the upper right hand corner of this, and I don't know if you can see my pointer or not. Hopefully you can. Who work with me, including Sarath, who's an amazing natural products chemist, Jenny, another chemical ecologist, and some of the postdoctoral researchers and other staff members who've been a big part of the team. This is our little field station down in Caribou Key in Belize, and you'll see a little bit more of that later. And just some of the various interns and students that have worked with us over the years. It's really been an amazing team of people and a real pleasure to work with them on some of these different topics that I'll share with you tonight. I also just want to also mention, since I'm not speaking very much about the Smithsonian Marine Station tonight, that we're up here in Fort Pierce on Seaway Drive. So I don't know, about a 25-minute drive, I guess, from Stewart, depending on how you decide to come this way. And we also partner with St. Lucie County on the St. Lucie County Aquarium. All the exhibits and educational programs there are run by Smithsonian staff, so members of the Smithsonian Marine Station staff. So we have a lot of different programming as well, and take a look at us online, and feel free. At the very end of the talk, I have my email address that I'll share with you, and if anyone has any questions or wants to reach out, please feel free to do so. So what I'd like to do this evening is tell you, as I mentioned, a little bit about marine chemical ecology. Some of you may be asking, well, what is chemical ecology? Well, I'll get there in a minute. I'll also share with you about marine cyanobacteria. A lot of people know them as blue-green algae. And yes, they can be nuisance species and cause all kinds of problems. I'll show you a few examples of that. They're also amazing in terms of some of the natural toxins that they produce. And we've been exploring some of those for anti-cancer activities, as well as looking at their environmental impacts. A little bit about a coral disease called black band disease that is caused predominantly by cyanobacteria, and continue that into the interactions between corals and algae. And finally, some more positive news, looking for chemical cues for corals and applying that to coral restoration. So first, what is chemical ecology, period, not just marine, but anywhere. And I think this example is probably one of the best that I could use to illustrate this. We know that many plants on land are toxic, and that those are often used for their chemical defense. They protect them from being eaten. Now, that, of course, can be inconvenient if humans or animals or something eat them accidentally. But they are the natural defenses for many, many different plants. As you'll see in the ocean, many things that can't run or get away or escape or don't have some kind of protective structures like spines or thorns or something. They can't run, they can't hide, but they can sure fight back with some very toxic chemicals. And this example of the milkweed plant and the cardiac glycosides, you've probably heard of those kinds of compounds, that they produce, like this one shown here, found in this is actually from oleander, but another butterfly plant. And these are very toxic compounds that can affect the heart and cause all kinds of palpitations and ultimately at high concentrations, death. So you're going to see a number of little chemical structures like this today. The backbone of the molecule, that as it's drawn, are all carbons. So everywhere that you see nothing shown here, these are all carbon bonds. And then you'll see the oxygens and nitrogens illustrated on these molecules. I know for some people who maybe have a lot of interest in chemistry, this is interesting and great, or pharmacy or pharmacology or anything like that. For others, you might be looking at them going, okay, but I think that it is interesting to illustrate the different kinds of molecules that we see in nature, and I'll try and talk you through them as we go through them. And these are a good example of the cardiac glycosides. So what happens is the butterflies eat the plant, actually caterpillars eat the plant. The caterpillars can sequester these compounds into the body of the caterpillar, and they are retained as the butterflies hatch out, and the butterflies are very toxic. And so this is a great example of how these might function as chemical defenses, because if a bird or something tries to eat a butterfly, in this case, that bottom picture, this isn't a great dinner time picture, but that bird is actually throwing up because these can also induce nausea and vomiting, even in humans. So these kinds of compounds. So they many animals quickly learn that these are not something that they want to eat. And as you know, many butterflies have these same patterns because it kind of reinforces this for visual predators that they don't want to eat these things. So now let's move into the ocean. That's the only plant picture I'm going to show you. In the ocean, probably the compounds we know best from the ocean are some of the marine toxins. This one is a classic. This is tetrodotoxin found in the pufferfish and other animals. And tetrodotoxin is very lethal. As you may know, people, especially in Asia, Japan, and other places, like to eat pufferfish. They have nice white flesh and are very tasty. But if aren't prepared properly, they can kill you if you eat them. And they have parts of the fish that have high levels of tetrodotoxin, like the ovaries and the liver and stuff. But they're not only found in pufferfish. These tetrodotoxins are actually found across animals on land too. Some newts and salamanders and things have tetrodotoxin. Back in the ocean, the blue ring octopus, famous for its lethal bite, also contains tetrodotoxin. And we discovered this this example up on the top right that looks like almost like a paper thin tissue. This is a flatworm, and flatworms are, just like they sound, very flat, very thin amount of tissue. But these guys produce a lot of tetrodotoxin, and what that allows them to do is they can engulf mobile prey like this cowrie snail, and release some tetrodotoxin, and that allows them to paralyze the snail and essentially just dissolve its flesh away. So this toxin is really, really important for that, for them to be able to eat some of the prey items that they eat. So a really, really fascinating series of animals that are all harboring these kinds of toxins. And then, of course, in Florida, we know a lot about the algal blooms that occur in our oceans from the Florida red tides that occur mostly on the west coast of Florida, but sometimes they wrap around the peninsula and come up on our coastline. I don't recall it happening for a real bloom in a number of years, but it does happen periodically. And these are caused by dinoflagellates, like you see pictured on the right there. And when the blooms get really, really dense, they turn the watercolor a reddish color, and that's why they're called red tides. These are producing compounds called brevetoxins that are very potent toxins, and fish kills can be associated with these blooms, as well as they can affect people, causing breathing issues and other things when it's at high concentrations. So those are some examples of toxins, but we know that these natural products can influence many other behavioral interactions, just like with the caterpillars and the milkweed or oleander or other plants. These can be predator deterrents in some cases. They can they can mediate these specialized behaviors, like we have with some insects that specialize on certain toxic plants. We can also see examples of competition that are mediated by the compounds, like between sponges and corals, or between cyanobacteria and corals. And these can also can have the compounds playing an important role. And those were just a few examples, but there's now literature supporting all of these different functions for these small molecules, these marine natural products that we sometimes hear about more for their biomedical activity than for their ecological activity. And all of these things, like defense and competition, anti-fouling activity, antimicrobial activity, just like many microbes produce antimicrobial compounds, protection against UV. Some marine animals have some great UV protectant compounds, and those are usually these small molecules as well. So there's a lot of different functions, and I'm going to kind of take you through some of them today. And I'm going to go back in time first, sharing with you some pictures from a long time ago. when I was an undergraduate at UC San Diego and kind of tell you about how I got into this field. And I've been really lucky that I was very, very interested from the get-go, and this is over 40 years ago, and been able to explore a lot of different aspects of marine chemical ecology and natural products over the years. Um, I got interested in diving early, and at UC San Diego, that was an easy thing to do. And I found myself in, I believe it was a work study program with Professor Bill Fennicle, who's now one of the senior spokespersons for marine natural products. He's really amazing scientist and has been active in the field for over 60 years, or about 60 years, I guess. And he, by working in his laboratory as an undergraduate, and I'm sure if there are any students on the Zoom call, you'll know that, having some of these experiences when you're an undergraduate student can be really amazing and give you a lot of opportunity to explore science. And I got really lucky to be working with him on these sea slugs called navinacs. And navinacs are pretty good sized sea slugs. They're 4 to 6 inches long. And they produce, they kind of follow one another slime trails. So they're very voracious predators, and they'll eat other sea slugs. So they're crawling along looking for food and following one another's slime trails. But if they're poked or prodded or alarmed in any way, they release these compounds that are shown at the bottom. And they're bright yellow compounds, but also apparently very good alarm cues. And so they'll immediately break trail and start turning away. And I mean, they're slugs, they can't go that fast, but they definitely do a about face and try to move in the other direction. So I had never heard of anything like this as an undergraduate. And the idea that these small molecules could play such a profound role in the behavior of these animals was just amazing. And so that launched my interest, and I've been doing it ever since. I decided to stay at Scripps working with Bill Fennicle for my PhD, and that was another amazing experience. I was there in the early 80s and got to explore a lot of different green algae. This is Halimeda. If you've snorkeled or dived on any coral reef anywhere, you've seen Halimeda. And Halimeda is an interesting algae because it's calcified, and it's pretty heavily calcified. It's up to, some species are 80, 90% calcium carbonate with a little bit of green to them. And so you wouldn't think they would be very tasty to fish, and they're not for many, many fish, but parrotfish and some of the other tropical grazers will eat them. And they are also producing these potent terpene feeding deterrents. And we were able to show that and show that they had some other functions. They have some antimicrobial functions as well. And I got to explore a lot of other different green algae and understand the terpenes that they are producing. And so terpenes are a class of compounds like you see here, carbon-based, no nitrogen, and very, very common in many land plants as well. After I finished my PhD, I got to move to the University of Guam Marine Laboratory. And I had actually done about 3 months worth of work collecting green algae for my PhD research when I was a graduate student. So I was familiar with the University of Guam and with Guam. And it was just an amazing, great place to do field work. It was a great place to try to develop my interest in some of these field assays to test feeding behavior and chemical defenses of a lot of different organisms, and right on the coral reef habitat. At the top of on the bottom picture here, so Guam is about as far away from us as you can go without turning around and basically flying back. The small little island out in Micronesia. Most people know of it because of the many military bases that are there, a big naval base, Air Force base. And it's a U.S. territory, and the phrase on Guam is it's where America's day begins because it's on the other side of the international date line. At the top of at the bottom slide there, you'll see the University of Guam perched on the hill and the little marine laboratory down at the base of the hill. So we were pretty much right on the water and could easily walk right out and over the reef and explore. And so it was a great place to do assays both in the field, and it's me driving a boat around, and in aquaria that were connected to an excellent seawater system, and just to have probably some of the best field access I've ever had anywhere. Uh, we were able to develop little kinds of assays to understand what kinds of defenses different seaweeds might have. These are green algae again. And we put them on these ropes and just hang them off a piece of dead coral rubble or something. And this is like a buffet line for fishes. So you've got all these different choices of different species of seaweeds here. And then on a coral reef where there's a lot of grazing pressure, the fish will come along, and you can actually watch them look at all these different algae on the line and maybe eat one or two of them and leave the rest. So they know what they're doing. They know what they like and don't like, just like we probably do at a buffet line. And it was a great way to figure out, mostly for my interest, what they don't like, because then they probably have some kind of defense protecting them, just like the land plants we talked about at the beginning. And then we could take this a step further. We could extract out the compounds. We make extracts of these algae, just like a solvent-based extract, like extracting them in methanol or ethanol or something like that. Think of it as like making your morning coffee or tea. You're extracting those tea leaves or coffee beans. And so we extract them. We can concentrate the extracts. We can purify the compounds, as I've shown you. And we can also mix them into fish food. We make like artificial diets out of auger and fish food, things that they like to eat, and then lace them with some of the compounds that we're trying to test to see if they actually deter feeding. So we could get right into the field assays and learn a lot about some of these chemical defenses of algae, and you'll see me applying this to some of the cyanobacteria later too. And what we've learned over the years is that how important these chemical defenses are for protecting macroalgae and especially benthic cyanobacteria from herbivores. And you can see some of the density of some of these grazing fishes in these couple of slides. The left one is from the Pacific and the right one is from the Caribbean, but these are all different grazing fish. They will definitely keep the bottom pretty clean when their densities are high. Now, there are a few more specialized grazers where we can see similar patterns to what we saw with some of the insects and the land plants. This is a little sea hare called Aplysia parvula, and it eats red algae, like the one on the right. That's Portiaria. And the Portiaria is laced with small terpene molecules that have halogenation. So they have bromine and chlorine incorporated into their structures, and they're quite deterrent to most fish and other things. But these little sea hares will eat away at them and incorporate the compounds into their bodies and into their ink. They spray out ink if you poke them. I think if anybody's messed around with a sea hare of any species, you know that. And the ink and the protections in their body are filled with these compounds. So they have the ability to sequester these compounds, just like the butterfly I was talking about at the beginning. And the other good example of that are these nudibranchs. Nudibranchs aren't herbivores, they're predators, but they eat sponges. And sponges are another group of marine organisms that are very well known to have chemical defenses. So here we have a little brightly colored nudibranch eating this gray sponge, and they incorporate the compounds from the sponge and put them into their body to protect them from grazers or from predators. And one more example, this one's kind of fun because this is a gray sponge. It's got cyanobacteria in it that live symbiotically within the sponge. And this brightly colored mollusk, a pistobranch mollusk on the right, it's similar to a nudibranch, but it's actually a different group of apistobranchs. And it's a kind of a mouthful, Sagamonopteran psychedelicum. And you can see where it might get the name psychedelicum from those patterns on its mantle, on its body. But this one is also sequestering very high concentrations of these potent sponge compounds called brominated diphenyl ethers. They're somewhat similar to some synthetically made compounds like flame retardants and things. And the animal, the Sagamonopteran, can sequester very high concentrations in its body. And we were able to show through some of our feeding assays that these are strong feeding deterrents to fish. Later, I worked with a colleague who's now at Georgia Tech. His name is Vinayak Agarwal. He's a professor up in Atlanta at Georgia Tech. And he was able to show that it's actually the cyanobacteria in the sponge, not the sponge animal itself, that is making these potent deterrent compounds. And he did some amazing work showing what the actual enzymes are that are making the compounds in the cyanobacteria and all of that. But so what makes this story even more interesting is that this is a tritrophic level incorporation of these compounds, because the animal is the Sagamonopteran is eating the sponge, but the compounds in the sponge are made by the cyanobacteria in the sponge. So it's a really super interesting relationship. So coming back to Florida in 2002, I was able to get this position at the Smithsonian, which was a great opportunity, and essentially to keep working at a marine field station. I've been so lucky to be able to work at a marine lab for my whole entire professional career, and to manage two field stations for the Smithsonian Marine Station here in Fort Pierce, where I'm based, and the Caribou Key Field Station in Belize, which is just a tiny little island designed for relatively short stays, 1 to 3 weeks or so, where you can have an intense field experience, because the island is surrounded on 3 sides by a beautiful coral reef, and then in the backside by seagrasses. And the whole area is just an amazing place to work, with real close proximity to all kinds of different tropical habitats. And then to come back and study Caribbean coral reefs, because I had done some Caribbean work when I was doing my PhD. So this was the first time in about 20 years I was able to do that. And I just like this picture because if you look at all the animals in this picture, a typical Caribbean coral reef with lots of sponges, and then these sea fans and sea whips, these octocorals, all of them. And, here's some algae. That everything I'm pointing to, everything that's so conspicuous on a reef is chemically protected from grazing. And also to come to Florida and find some of my favorite marine algae, which I know most people don't like these things, but I just think they're super cool, are these blue-green algae. And at one time, most of these are examples of ones that live on the bottom. They form these filamentous mats. It's almost like thick hair. And at one time, they were almost all called Lingbia. So if you've heard the term Lingbia mats, etc., that's what we're referring to. But we now know that the taxonomy is much more complex. I'll show you in a few minutes. But they form blooms all around the world. Florida seems to be very, very prone to blooms of blue-green algae. This sign, though, down at the bottom is from Queensland, Australia, where there was a big bloom. Now, some of our best-known blooms in Florida are these planktonic blooms of microcystis. This is, unfortunately, from the St. Lucie estuary down near where you are in Stewart. And this is a microcystis bloom. Microcystis is a very toxic blue-green algae. These blooms often start in Lake Okeechobee. They almost always have blooms in Lake Okeechobee in the summer months. And then when freshwater releases are necessary, and these things are released out to the coast, then they can get into the estuaries as well. This is just one of those really amazing super blooms. But here are some other pictures of microcystis blooms in Florida. These photos are from not only the Stewart area, but St. Johns River up near, up closer to Jacksonville. They unfortunately were somewhat plagued with these algal blooms. When I first got here, shortly after I got here in 2002, from about 2004 through 2005 or 2003 through 2005, we had these really dense mats of cyanobacteria on some of the reefs off of Broward County, around Pompano Beach, Fort Lauderdale. And you can see these are kind of a reddish color with overgrowing everything. The mats were so thick. And what you see at the bottom right is a microscopic image of what these filaments look like. They almost look like a coin roll where all the little cells are wider than they are tall, and they're all stacked into this polysaccharide sheath that the filaments are growing in. The really interesting form in these benthic blooms. And the compounds they produce are these compounds called protease inhibitors. There are a number of different types of protease inhibitors. Some are inhibiting digestive enzymes like chymotrypsin and trypsin, and it is thought from some freshwater work, because these are microcystis and other freshwater cyanobacteria also make these kinds of compounds. It is thought that they are inhibiting digestion by grazing grazers, like planktonic grazers as well as benthic grazers. And so that's why so many of these cyanobacteria might produce these kinds of compounds. But here are just a few more pictures, mostly from Florida. These are some seagrass blooms that we worked with in the Indian River Lagoon. And some of the, this is that same bloom off of Fort Lauderdale down in the Florida Keys. And a couple of these are from Guam as well. So what happens, what fuels these blooms? And how do we prevent them? This is something that we've been considering quite a lot in Florida. And there are unfortunately a lot of easy ways to promote the blooms and not so many easy ways to prevent them. Nutrient enrichment promotes algal blooms, just like, it helps your grass grow if you add nutrients or any plant that you fertilize. The other thing is the warming of seas. Cyanobacteria really like warm water, and so they'll bloom pretty regularly. And then the toxins deter the grazers, so that things don't want to eat them. So there's no, what we ecologically call top-down control. There's nothing preventing these things from growing, or not very much preventing these things from growing, and other things that stimulate their growth. So it's very, very challenging. Takes nutrient control measures. It's much harder for us to control the temperature of the water, but prevention is often key to success in controlling these blooms. And when I was in Guam, I first started working with Hendrik Lusch, a longtime colleague now. We've been working together for probably almost 25 years. Hendrik moved to the University of Florida right around the same time I moved to the Smithsonian Marine Station. So we both ended up in Florida at the same time. But we had started to work together in Guam when he was a graduate student at the University of Hawaii. And we found quite a number of really interesting and very potent toxic compounds in cyanobacteria. And Hendrik is in the Department of Medicinal Chemistry at the College of Pharmacy at the University of Florida. And our interests are very complementary. We're very interested in the discovery, some of the taxonomy, the chemical ecology of these blooms. And he's very interested in the compounds and what they might do biomedically. So we've had some long-term support by the National Cancer Institute to explore that. And we have a grant now together, and also have worked together on a number of other projects. So this has been a really fruitful collaboration, and has led to a few potent cytotoxic compounds that do kill cancer cells. This one, aprotoxin, he has continued to work on since he first discovered it as a graduate student working together with us in Guam, because Apra Harbor is the main harbor on Guam. Largazole came from Key Largo, one of our first discoveries when we moved to Florida. And these are both compounds that he continues to work on in his laboratory. They've done a lot of synthesis, made a lot of analogs of these compounds because of their potent anti-cancer activity. This is another one that's really interesting from that perspective, Caldora. Caldora is one of my favorite cyanobacteria because it can be very diverse, at least in terms of its color and a little bit about its texture and morphology. But it's kind of like giant blobs of cotton candy on the reef, almost, if you look at the more pink ones. And it is one of the most toxic, lethal things ever. Uh, Needless to say, probably not a lot of things like to eat it on the reef. And we described this. This wasn't really very well understood taxonomically, and so we described this species as Caldora peniculata. And these are the toxins that I'm talking about. Largazole that I showed you earlier, again coming from collections of this from Key Largo. But these other compounds called dolastatin and simplistatin, and these were, we discovered simplistatin, but dolastatin 10 was already known. And dolastatin 10 is really interesting. To me, this picture really exemplifies that cotton candy-like texture of this stuff on the reef. Dolastatin 10, analogs of that compound are currently in anti-cancer or components of anti-cancer drugs that are used today. So these are very toxic molecules and very, very interesting to find them around the world. We found these in the Pacific, in Guam, in Florida, and other places. So super interesting cyanobacteria. I wanted to show you one picture of a former postdoc of mine. He's now back. He's a Swedish gentleman, Nicholas. He's back in Sweden now, working for the on algal blooms and other things for the Swedish government. But he was very, very interested in some of the taxonomy of these, and this we worked together really well on describing some new genera of cyanobacteria. And these are a couple pictures of a genus called Okeania. These had all been thought to be part of the overall clumped in with Lyngbia. And now we know that they are pretty distinct genetically. And the really fun thing, I know this is a very busy slide, but as he started to work out and better understand the biodiversity of these algae and use phylogenetic methods to group them into different species, what we discovered was that once you started to make sense out of their biodiversity, out of what the actual species are, and he named all of these working with us here at the Smithsonian Marine Station, we found that every species had its own suite of compounds. And I know these are, all a bunch of, chemical structures here, but I think you can see that there are different structures for each species. And so this was really kind of a new revelation because when everything was grouped under the genus Lyngbia, the thought was that, Lyngbia was just producing all these different compounds. But what really was the case was we didn't understand the biodiversity of these cyanobacteria, and we still don't. There's a lot of work being done. I have a colleague who's down in Davie, near Fort Lauderdale, Dale Laughinghouse, who's continuing to work and understand some of the some of the systematics and taxonomy of these cyanobacteria. And finally, this genus Dapis, I got to show you because this was one that you see in the Indian River Lagoon and around Florida all the time. And it forms these big floating mats in the summer. So if you're out boating in the IRL and you see these big mats of what look like pond scum, slime floating around in the water, it's probably this species Dapis pleosa. Dapis is also found in overgrowing coral reefs throughout the Caribbean and around the world. And it's just a really, really interesting genus. So with all these different cyanobacteria, having favorable growth conditions and growing prolifically in Florida and other parts of the world, in tropics and subtropical habitats, what we start to see are more interactions between corals and the cyanobacteria. And so that's going to be the next sort of topic of my talk is some of these interactions and how cyanobacteria, which are favored, by warm water temperatures and other things that are really negative for corals, how they can proliferate and bloom. And this is just a couple of examples. This is from the 2023 warm, bleaching event that bleached many, many corals throughout Florida and in other parts of the Caribbean too, including some of these octocorals that turn stark white. So bleaching is when the corals lose their symbiotic algae. So they go from a golden tan color to white color because all of the algae are essentially thrown out of the symbiosis. And it can kill corals. In this case, this one is probably not going to survive because it's been overgrown completely by cyanobacteria. And I have this picture I wanted to show you because we were at the time tagging and following individual coral heads down in the Florida Keys because some of these had a disease that's known as stony coral tissue loss disease. It's plagued Florida since 2014 and killed many thousands, perhaps hundreds of thousands of corals. But we were watching these and monitoring them for disease. But what we got caught instead was this bleaching event. And I just thought this was really interesting to show you because this is the same coral over time from May to December. And you see it's got this, some of the color variation here is just people using different cameras on the different collection days. But you can see that it stays at a kind of a golden brown color until when the water got really warm at the end of July. And then by August, look at it, it's stark white. No, almost none of the symbiotic algae that gives it its golden brown color. And then what's interesting, it stays white through September, but then in October, you start to see some of this golden brown color coming back right around the edges. And it could be that some of it continued to live down here where it was a little more shaded, or they're beginning to pick up symbionts from the water column. They can do both. And you can see by November, it's recovering. It's getting more and more brown, a little bit of white bits here and there. And by December, it's mostly recovered. And I just thought this was a really good example of how one group of organisms can really proliferate and take advantage of these algal blooms and these warm water events. Whereas another group, and unfortunately we did hear about many corals being lost during that 2023 bleaching event. So one other interesting thing, kind of following up on things that can harm corals, is some of the diseases. And this is one that I thought fit this theme of cyanobacteria because this is a cyanobacterial disease. It's called black band disease because you get a black band growing here on the coral. And this is a cyanobacterial mat that is growing right into the coral tissue and killing the coral behind it. So this mat will progress across the live coral tissue. Again, this is that nice golden brown color that we see on corals. This is a different species, kind of a slightly different shade of brown with also a cyanobacterial mat. And this will eventually kill the coral. And what's left behind here in this case is dead coral skeleton. In the bleaching I showed you, the tissue was still there. It just didn't have the symbiotic algae, so it was white. But here, this is killing the coral. So this is not coming back. This is dead skeleton. And this is a cyanobacteria called Rosea, and it's found in black band disease around the world. But there's also bacteria and other things in these mats, and people are still not entirely sure what kills the coral. It's thought that sulfur bacteria at the base of the mats might be causing anoxic conditions or toxic conditions that kill the coral. But from our chemical perspective, we were really interested in seeing what was in these black band mats. No one had really done a thorough study of the natural products. And so the first step was to try to collect it. And we spent a lot of time doing this. We spent years collecting enough black band mat in these syringes. That was the only way to get it off. It really adheres very well to the coral and to the coral skeleton. We kind of slurped it off with these syringes and got enough that we could finally characterize these compounds. They were new. They were never previously described in nature. And we called them the leukeolides because we collected so much black band disease at Leuke Reef that we could, it was one of our predominant collection sites. And we don't, to this day, know exactly what these are doing in the mats, but we've looked at other species of Rosea that don't grow on corals and they don't produce these compounds. So it's playing some function, probably important for overgrowing corals. And what we do know is that it can help reduce reactive oxygen species. So it's kind of it has antioxidant properties, and that might help it or facilitate the growth of the mats on the coral, because coral, like many plants, one of its first lines of defense against predators is to produce reactive oxygen species like hydrogen peroxide. That is a chemical defense. So we are still very interested in these compounds and what they're doing on reefs. And I want to just wrap up with the last couple minutes of my talk about some of the more positive things that we can do with chemical ecology and understanding reefs. And that is how we can help with coral restoration. And if you look at these two kind of scenarios for what a reef can look like, this is, of course, a nice healthy reef with coral and fairly bare skeleton, fairly bare substrate, not a lot of algae growing on it. And then this is one where we're looking at on the right is a more degraded reef. And ultimately, we want to be able to bring some of those more degraded reefs back to life and look more like the one on the left. And a lot of people are working on this. A lot of people, as you know, in the Keys and even in Southeast Florida and Broward County and other places, Miami, are growing corals and putting them back out on the reef and trying to make a real difference there with reef restoration. But we were more interested, and a lot of people are getting interested in this now, in the larval stages and how by tapping into the sexual reproduction of corals, we might be able to facilitate some more diverse restoration options. And so this is a coral called Porites estrioides. It's actually a brooder. Corals can spawn or can reproduce sexually a couple of different ways. But these brooders release fully developed larvae that are ready to settle and metamorphose. And this looks like what one new coral spat looks like, one little settler. You can see the polyp. It almost looks like a little sea anemone with its mouth and the tentacles around it. And so here we have four polyps of Porites, and then they eventually grow into a small juvenile and then back into an adult. And so these larvae allow us to understand what facilitates larval settlement. The spawners, most of our corals are spawners, and they're very similar. The biggest difference is these brooders release these larvae. They're completely ready to settle and metamorphose. But the spawners release sperm and eggs into the water column, and those have to fertilize and develop for 3 or 4 days before they're ready to settle. So a lot of our work's been done at our little field station in Belize, which is amazing. Like I said, an amazing place. You can see the underwater reef there. And this is what an Acropora palmata spawning event looks like in Belize. And it's like being in a snow globe. All those pink dots in the water column are coral larvae, bundles of sperm and eggs that are being released by these corals. And so we're able to catch them essentially under these little nets that we put out. You can see on the right the little pink bundles floating. These are mostly eggs of the coral, and they're really lipid rich, so they're full of fats and they float. And then we bring them back. We fertilize them in the lab, and then we bring them back and grow them in these little tubs for 3 or 4 days. And then we have nice larvae that we can work with. And then going back to some of our interests with cyanobacteria in competition with corals, we decided to look at that, how different algae and cyanobacteria might inhibit, in some cases, or facilitate larval settlement. And so we have some algae here. This is a brown alga called Dictyota, and little chunks of rock with coralline algae, crustose coralline algae growing on them. And then we put them inside these little chambers so the larvae can't get out, but water can flow through. So they are plastic chambers, polystyrene chamber or polyacrylic chambers with mesh on both sides. That the mesh is smaller than the larvae, so they can't escape. And we can put these right out on the reef and get as close to natural conditions as we can to study their behavior. And here you see some results with our Caldora, the one that I told you produces these really toxic compounds that are biomedically important. And if we look at the settlement, if we just use plastic algae as a control, and then one species of brown algae, a couple cyanobacteria, these Acropora palmata really don't like to settle on the Caldera, which is not, or near the Caldera, which is not too surprising. And then these brain corals, Pseudodiploria strigosa, really don't like this other species of cyanobacterium called Hormothamnion. They don't like Caldera very much, but this one wasn't statistically inhibiting settlement. Now, we do know that there are some things that stimulate coral larval settlement, and that is mostly these species, different species of these coral and algae. They look like pink paint on rocks, so they're just thin encrusting red algae that overgrow rocks, and it's been known for some time that coral larvae and other invertebrate larvae will settle on them. And so we decided to tease apart some information on which species the coral larvae, which species of coral larvae like to settle on which species of crustose coral and algae. So we were able to tease some of this out all from our work in Belize, looking at the patterns of settlement for 7 different Caribbean corals. These are the little larvae when they're elongated like this on the left and exploring the surface of the coral and algae. And then on the right, you see 3 beautiful new polyps of brand new settled baby corals. So it's pretty fun to do this kind of work because you are really, bringing things to life and understanding the patterns of what really facilitates the settlement. So now that we know what species are abundant, we use them, or are good inducers of coral larval settlement. We use them in a lot of different experiments, and we've been trying to get better understand what some of the chemicals are. We can also put settle the coral larvae on little settlement plates and put them out on the reef. Check how they do. Unfortunately, it takes a lot of coral larvae to rear some adults because it is a very vulnerable stage of their life history when they're little settlers like that. About a decade ago, I started working with Jennifer Sneed, who works here at the Smithsonian Marine Station with me. And I believe she gave a talk last year. Some of you may have heard her talk about some of the work with one small molecule that we isolated, probably back in 2014. So that's over a decade ago, that comes out of bacteria that live on coral and algae. And this is a molecule called tetrabromoperol, or TBP. And we showed that this could induce settlement of coral larvae and that many different coral larval species settle on it. So Jennifer has been really pushing hard and working hard to better understand this. We know now that this is a very specific compound. If you start to remove, we've worked with some chemists to synthesize some analogs, start to remove bromines, replace bromines with chlorines. It doesn't work. So it has to be this molecule. This is what the corals respond to. And so we're starting to work a lot with, Jennifer's been providing TBP to a lot of different coral restoration partners who are starting to use this molecule for larval settlement. So there are a lot of implications for understanding these chemical cues for coral restoration. First, we know that chemical cues are important, that they're perceiving certain species of crustose coral and algae, different molecules on the surface. The smell of the reef may be critical for recruitment success. This has been suggested for fish as well. Little larval fishes find the reef by sound and smell and other things. We know that certain species of crustose coral and algae are particularly effective at inducing larval settlement, and that some of these macroalgae and cyanobacteria can deter this. So, all of these things are working together to either promote or inhibit coral larval settlement. And our latest work is now focusing on compounds that are being released into the seawater that can be cues. And so there are a lot of challenges working with that because rather than we have to be able to retain the compounds back out of the seawater to be able to study them and figure out what they are. So that's kind of been our latest project for the last couple of years. So I want to wrap up and finish up. It's 50 minutes, and I think everybody's probably saturated by my discussion about chemical ecology. But just kind of reiterate or to review what I've talked about. I've talked about sea slugs and uh, their ability, slugs and their ability to sequester some of these compounds, how many generalist consumers are deterred by these compounds that many benthic organisms on the reef produce. Slime, I'm an odd one, but I just find these cyanobacteria fascinating. And the more we explore them, the more we find novel compounds and novel behaviors. It's just unbelievable. And then I didn't talk about some of our other coral disease work, but I did talk about black band disease because it fits into this topic. And it's just a really fascinating the whole subject of coral disease is just fascinating and also really important to get a better understanding of. And I want to acknowledge not just my team, but my many collaborators. I talked about Hendrik and his group, some of our coral research collaborators that have been amazing to work with and we have ongoing projects with. And I just really thank you for your attention. It's been 50 minutes and I hope you enjoyed it, and got to thank all of our different funding sources that have supported this work over the years. And happy to take any questions. And there's my email. If anybody wants to jot it down or anything, feel free to reach out, and I'm happy to talk to you more. And of course, share with you a little bit more information if you're interested. And of course, come and visit our aquarium when you have a chance. And I think I'm ready for questions. Thank you.
All right. Awesome presentation, Val. We had, believe it or not, 250 plus people in attendance tonight. So I'm glad we pivoted. We were originally going to just cancel the presentation, and it was fantastic that we were able to still do some sort of program, even though it's a little bit different than what we had originally planned. Let's see here.
So I'm going to stop sharing, right?
Yep. Yep.
Zach. And if anybody missed this or wants to get hold of me, you can just ask Zach. He can put you in touch with me.
All right. So we've got some questions rolling in already, but I would encourage all of you, if you have any questions at all about any aspect of Val's presentation tonight, go ahead and throw it in the Q&A box and we'll try to get to some of them. My first question relates back to one of your intro slides where you showed the butterfly being eaten by the bird. Somebody wonders whether agamas are sensitive to toxins like that. This viewer has seen them eating caterpillars, and they're wondering if the agama lizards are potentially impacted by secondary metabolites or whether they're immune to them.
Yeah, and I don't really know the answer to that, but it's an interesting question. Of course, those agamas are invasive, right? So they probably hadn't previously encountered any compounds that would be in our Florida caterpillars, but most things are, I won't say most things because it does vary, like pufferfish, can sequester all these compounds and other things too. I don't know, and I've often wondered if those agamas have any kind of chemical defenses themselves, because they have that bright coloration, which is often associated as a warning coloration with chemical defenses. So I don't know the answer to that, and if anybody knows, please write it into the Q&A. Some of our fish, wildlife folks that have more familiarity with wildlife or invasive species might know about those guys. But yeah, I don't really know the answer. I know they're generalist predators, and most generalists are, somewhat avoid toxic foods. So, they can get around it a little bit by a mix and match diet, which is certainly what I've heard that agamas eat, that they're eating, eggs of other lizards and plants and whatever. They're pretty generalized. So they're probably in grazing plants, they're probably getting some toxic compounds. And so do we, right? I mean, we eat lots of plants, and broccoli and all of those have compounds that are protective. They have some phytoprotective properties. But a diverse diet for an omnivore is really important.
I mean, how about tobacco, right?
Tobacco is, well, of course, we use it probably mostly not to eat, but that is certainly a classical feeding deterrent for most insects. It hasn't kept humans away though.
So our next question is about cyanobacteria, and I'm not sure whether the participant is talking about Microcystis or Lingbia dapis, but the question is, do some of the coastal cyanobacteria produce neurotoxins that affect humans? I read somewhere that they were suspected to be a causative agent for ALS.
So that's a really interesting question. I think what they're referring to are, is it BMAA, beta methyl amino acids or whatever they are. Um, and I didn't talk very much about those. They are actually kind of tricky to analyze. The quantitative analysis for those compounds is somewhat specialized. And they are pretty common in different cyanobacteria, though, both the benthic types that I showed you as well as the some of the planktonic types. So um, those are have some neuro toxicity, I guess is the way to put it. And so they can be pretty bad. But I mean, microcystis is bad all around because it also produces these microcystins, which are liver toxins and give you liver cancer. So, you know, if they don't get, they don't get your liver first, they might get your brain. I mean, they're just not good in general, some of these blooming species.
How about Lingbia slash dapis? Do we know the potential implications of exposure to those types of cyanobacteria on human health?
The main compound that's been studied that way is one called lingbiotoxin, and uh, it has it is toxic, but it also causes a contact dermatitis in a lot of people. So it gives people what they call swimmer's itch. If you get it, if you're susceptible to it, if you get some down like your bathing suit when you're swimming or something like that, it might cause some irritation. It, it Most people wouldn't eat those benthic cyanobacteria. The problem with the planktonic ones that I showed you, like the Microcystis bloom, is they are freshwater cyanobacteria and they get into water supplies. So it's causes a lot more problems for water treatment plants and things to have to get those compounds out. And when they aren't removed, that's when they start to impact humans.
All
Most
right.
people are not purposefully consuming them, right? But sometimes pets can consume them in lakes. I've heard of cows drinking them and getting sick, or and dogs getting sick. So you do have to watch your pets around any lake that's got an algal bloom.
So our next question, the molecules that you showed us tonight look very complex. Do you determine their structures yourself, or do you collaborate with chemists?
So we have in working in my laboratory a really good natural products chemist, Sarath. I pointed him out at the very beginning. So we do some of our own structures. It's a combination. We do some of our own. We partner with other people, um, and so. But yes, we do have the capacity. We've characterized several dozen compounds here in our laboratory.
All right. Is it possible to do something to stop the black band disease before it kills the coral?
So people have messed around with a lot of different ways of killing it for decades. I like to think that us slurping it off helped some, but we couldn't remove every last filament. So I'm sure, we just slowed it down a little bit. One thing that had worked, it's somewhat labor intensive, um, is that you can put like clay, like modeling clay or something, or epoxy with chlorine in it over the band, and that kind of kills it or slows it down. So there are some manual treatments that people have tried that sort of work. It's just kind of labor intensive, right? So it takes a little bit of uh, person power to go out and apply those. People are still, Erin Muller and her team down at Moat Marine Lab are still working on ways to try to, stop or slow down the disease. So there are definitely some treatments still being applied.
All right. Boy, we've got some great questions coming in. I encourage everybody, type them in the box. We'll keep going as long as we can here. We don't have the library turning the lights off for us tonight, so we can stick around and get
That's
all your
true.
questions answered. All right. So can you expand on the influence of increasing marine water temperatures beyond coral bleaching? For example, decreased oxygen availability, the promotion of the growth of bacteria that produce hydrogen sulfide, etc.
Yeah, all of the above. So we've been doing some work here. We had a really talented postdoc help set up some aquarium systems. They're fairly large tanks, about 50 liters, and each one can independently control oxygen, pH, and because ocean acidification is another impact of rising CO2 and temperature. And so she's been able to do some manipulations, mostly with corals. And needless to say, deoxygenation isn't good for most organisms. You get below a threshold of about 1.5 to 2 milliliters of O2 per, what are the units? Whatever, milligrams of O2 per milliliter. And I might be wrong about that, sorry. But anyway, you start getting into hypoxia or, really physiological impacts on the organisms. And so we've worked with corals, coral larvae, and other things. Larvae, surprisingly, were fairly resistant compared to some adult corals to lower oxygen. I mean, nothing's going to survive when it is completely depleted, but they can survive at around, 1.5 to 2.
And to put that into perspective, that's much lower than something like a fish could survive in.
Depending on the fish, right?
Yeah.
Yeah, some of the bottom dwellers are probably more adept at handling those lower oxygen levels. And the thing about some algal blooms, like for instance up here where we are and north of us in the Banana River, we've had a number of brown tides, which are Aria umbra and other weird brown algae, various planktonic brown algae. And those can kill fish and other organisms without toxins. They just, when they really bloom up and crash, then you get low oxygen in the water and things just die because of deoxygenation. So it's not just toxins in algal blooms that can kill things. Of course, toxins can help that along, right? So yeah, different algae can really have different mechanisms of being harmful.
Our next guest would love an explanation of the process that corals go through from the larval phase to the polyp phase. They're wondering whether there's a metamorphosis involved, like a caterpillar metamorphosing into a butterfly.
Yeah, absolutely. If you, I don't know if let me see if I can just go back real quick because I think the one picture of it is pretty good. Let's see, are my slides up, Zach?
They are loading. Yep.
Yeah. Okay. So let's just go up a couple slides because I showed you some. Oops, this one I think really shows it. So this is essentially metamorphosis, right? You're going from a larval form, which is very different morphologically. Think of this sort of like your caterpillar in a sense, right? It looks like a blobby sort of thing, not a lot of morphology to it. It's called a planula larvae. It's a type of larvae that many corals and related organisms have. And so this going to this one single polyp stage where it looks almost like a little flower petal. It's almost like a little sea anemone, right? You've got the tentacles surrounding the mouth of the coral here. And this is metamorphosis. So yeah, maybe this one's not going to fly away like a butterfly, but you've got the same process going on here, going from a larval stage to what people call spat. And spat's a term that we often hear with oysters too, right? Oyster spat are the new little recruits that are also essentially a metamorphosis from a baby oyster that floats around in the water to a settled oyster. And we see that in the Indian River Lagoon all the time. So it's a really, really interesting feature of many different benthic invertebrates. They have a larval form that's up in the water column. It can swim. It's sort of designed for dispersal. And then they have the benthic form. And so the critical step, one critical step that we don't understand that well is how do they find the right place to settle? right? Because they're going from a larval stage where they can swim around, explore, to a benthic stage. And once they commit to settlement, they're pretty well committed. Like they're not, it's not it's off. There are a few cases where maybe it's reversible, but not very many where you're not going back from a settled coral baby to a larvae again. So it's a really interesting process that's very much like metamorphosis on that we're more familiar with.
Great. All right. Somebody wonders, with all the toxic compounds you talked about tonight that are found in cyanobacteria, why do some health foods, in quotes, list blue-green algae as an ingredient?
So the main one that's grown for health foods is one called spirulina. It's a really cool one. I wish I had a picture of it. I don't. It's like a little corkscrew shape almost. So it's the same filaments, but they grow in this sort of curlicue corkscrew. Uh, Supposedly it's not toxic. You know, the risk with some of the cultures, I would the biggest risk. I mean, some of them are cultured in very sterile conditions. Some cyanobacteria and other algae and other planktonic algae are cultured in big ponds. So there, I think the biggest risk might be contamination if something else got in there that was producing toxins. Right? They might not. Yeah, it depends depending on the testing and things. That's one thing about many health foods and supplements is they're not as rigorously tested as for instance, FDA approved drugs or something. So contamination might be one concern, but yeah, they're clearly using species that are not producing a lot of these toxins.
All right. I'm going to roll a couple of questions into one because there's some overlap. Are there other medical uses of marine toxins in addition to cancer treatments? And how has your research impacted drug development or treatment protocols in medicine?
Yeah, let me start with the last question. I think that I have contributed to that sort of research, but that the people like my colleague Hendrik Alouche, who's a real medicinal chemist, has done a lot more. He's often taken the drugs that or the compounds, not the drugs, the compounds that we've discovered, and really worked very hard and systematically to understand their mechanism of action. Like, not only do they kill cells, cancer cells or bacterial cells or whatever, but are they actually, what are they actually doing to mechanistically inside the cell to do that? And we know, for instance, that our compound largazole, this is all through Hendrik's work, that our compound largazole is a histone deacetylase inhibitor. And that means that it helps unwind the DNA. The DNA is all packed up in our cells, right, in our nucleus of our cells, helps unwind it, which can then allow it to begin to make proteins and all of that more readily, and it inhibits that process. So it can help inhibit cell growth that way. Others inhibit mitochondria. Others bind microtubules, which are really important during the cell division stage. And many of our cancer drugs, like Taxol, the one from the yew tree, the breast cancer drug, is a inhibitor of microtubules. So we found one or two that do that. And some have some unique mechanisms, but he has really worked really hard. So I like to feel like I've helped and collaborated, but I can't really take credit for that really excellent cell biology and everything that they do.
Right. Tonight you showed a couple of different kinds of cotton candy looking cyanobacteria that are smothering the reefs. Are there any specialized grazing animals that are able to control those cyanobacteria?
So the only thing I know that eats cyanobacteria is another little sea slug. I showed you that sea slug at the very beginning that eats the red algae. There's another species of sea hare called Stylacylus, and it's found around the world. And it is a specialist on cyanobacteria. There are also a few smaller snails that we found in those mats that are specialists on cyanobacteria. So those are really interesting. And I don't know that they're numbers could ever get great enough to truly control some of these blooms. But certainly before they bloom, when they're just, these are natural components, just like anything that blooms, they're natural components of the ecosystem. It's when environmental factors or other things, too much nutrients, too little oxygen, other things, environmental challenges can knock out a lot of the competitors, allowing these algae to really reach vast populations, right? So that's what a bloom is. So they're there naturally. They're part of the ecosystem. I'm sure sea hares play some role in controlling them at that stage. Once you get to the real bloom stage, I'm not sure you can get enough sea hares to take care of them, but they definitely are playing some role. They're one of the few specialists, some of these different mollusks.
All right, that's a good lead into our next question. You just talked about changing environmental conditions and how that might impact blooms. This guest wonders whether changing environmental conditions may render organisms more sensitive to the toxins you talked about tonight.
That is a really good question, and I think the answer is yes, but there's probably not a lot of evidence for it. One of the things about coral bleaching is that when they bleach, they're getting stressed, right? Because that symbiosis between corals and their algae that lives in their tissues is very beneficial for the nutrition of the corals. The algae are photosynthesizing just like any other plant like organism, and that means that they're providing some of those carbohydrates to the corals, and the corals get a lot of nutrition from that. So when they get a bleaching event and most of the algae are gone from their tissues, most of them survive. All corals will, have the symbiosis part through their and get some nutrition from the symbionts, their algae, but they also feed just like a sea anemone or anything else. They feed on phytoplankton and zooplankton that come by. So the only way that corals really get through a bleaching event and recover is by feeding, because they're not getting as much of their nutrition from the plant, from the algae in their tissues. So that is one important factor. And so anytime an organism gets stressed like that, there have been a number of studies. We haven't actually worked on this, but there are quite a few studies that show that the next year or so, they could be more susceptible to disease, or they might not have as much reproductive output as they did the year before in terms of producing more, get eggs and sperm. So they're definitely impacted by these environmental stresses, big major environmental stresses, probably even minor ones, and that can have an impact on them and probably make them susceptible. Knocks out their immune system. I mean, just like us, right? If you get really chilled and then exposed to a cold, you might be more likely to get it than if you were super healthy and not feeling stressed.
All right. Even though the library is not going to be kicking us out here in a few minutes, I would still like to wrap up by 8. So I think we have time for maybe one or two more questions. What is the typical?
I see one. Oh, I just wanted to mention, Zach, I see one hand raised. Do you see that in
I
your
think
view?
that popped up at the very beginning of the
Okay,
presentation. Has
all
just
right.
stayed up the whole time. Yeah.
Okay, all right.
So what is the typical rate of progression for black band disease? And are there any obvious factors that expedite its growth, like water temperature?
Water temperature expedites its growth. Enhanced nutrients expedite its growth. Josh Voss at FAU did some work on that for his PhD research. He worked on black band disease a number of years ago. And so again, some of these environmental impacts that can fuel plant growth and algal growth in general can fuel black band disease too.
If you don't mind, I want to ask you a question.
Sure.
We'll wrap up with a Zach question. In
Okay.
in parts of the Indian River Lagoon system, particularly the Banana River, we've seen seagrass being replaced by a type of macroalgae, Caulerpa prolifera.
Mhm.
Is that species of algae chemically protected? Do we know anything about the chemical profile of Caulerpa prolifera and whether it's a, a type of algae that isn't prone to being grazed by herbivores?
Yes. So one of the few grazers for the Caulerpa are the sacoglossans, right? The little cute little green sea slugs that can eat all the green algae. So yes, that was one of when I worked on my green algae for my PhD. Caulerpa was one of them, and all the caulerpas produce these interesting terpenes. I showed you the Halimeda terpenes, but the caulerpa terpenes are somewhat similar. They're a little smaller. They're 15 carbons instead of 20 like Halimeda compounds, but they are yeah, interesting. They can deter feeding by fish, and we don't really know everything they do. They might have some interesting uh, impacts on competitive interactions with either seagrass or other algae. We just don't know. We suspect some of those might be going on. So there's some really interesting projects there, I think, with the Caulerpa prolifera.
All
It
right. Well, I think
stabilizes the sediment, so it's considered kind of a good thing in that regard, but it's definitely replacing, in some areas, replacing seagrasses with these algae.
all right. Well, I think with that, we're going to wrap up. One of the downsides of not being at the library is we don't get to hear a couple hundred people clapping. So I think the two of us here will give you a rousing round of applause. Three if you count our cat. But we really appreciate you pivoting tonight. I know this is a disappointment not to be in front of a live audience. I will tell you, even with this wonderful, almost 30-minute Q&A conversation, we still have well over 100 people logged in and watching. And that's a testament to how these presentations can turn into conversations. And I really appreciate you taking the time to answer everybody's questions tonight. We got through almost everyone's questions. And I just want to remind the remainder of you who are here tonight to please join us in a week. Justin Perrault from Loggerhead Marine Life Center is going to give an amazing presentation with some really good imagery all about the sea turtles that nest on our local beaches. Thank you again, Val. And I'm sorry we had to cancel our in-person presentation, but I think this is still a wonderful opportunity for people to learn about your work. And I want to thank everybody for participating and have a great night. Bye, everybody.